My name is Jung, Lee Tian Jung, from a molecular biologist at the Agricultural Genetic Institute in Hanoi, and today I would like to introduce to you very briefly how we have done with the lamb to detect cassava switch broom diseases. Okay, so here we are in the ninth floor of AGI, and our lab is International Laboratory for Cassava Molecular Breeding, and we go to see a few persons who are working on cassava. So now we are in the lab, the molecular biology lab, and this is a Queen, this is a Nam. We are working on together. We are working on cassava pests and diseases.
Hi, my name is Queen. I'm a master student at Hanoi University of Science, and now I'm working on cassava lectin project.
Hi, I'm Nam. I have finished a master thesis from the Hanoi University of Science. My research to develop the lamb assay to detect phytoplasma causing cassava with broom disease.
So as you know, cassava Swiss broom disease is very serious in Southeast Asia and Vietnam as well. We call it cassava Swiss broom because when infected into cassava, they make the plant look like a Swiss broom. So here you can see in the screen. Swiss broom disease is spread all over Vietnam from the north to the central highland to the south on the cassava production in the country. When infected with cassava Swiss broom diseases, the productivity reduced seriously, and also from the harvest, the starch content also reduced greatly. And research from colleagues around the world has shown that the pathogen causing cassava Swiss broom diseases is a phytoplasma. You can see on the slide. And this phytoplasma can be transferred from diseased plant to a healthy plant by an insect vector. And there is other research that's going on to find out which vector, which insect are helping transmitting the disease from diseased plant to healthy plant. So there are some various methods for disease diagnostics in plant as well as in animal. And there is a method called LAM, that means loop mediated amplification of nucleic acid, and this method relies on the formation of the loop at the umbilical, that where you can see the principle right now here. This is the cycling amplification step of the method. So next. Next, it's more amplification. You will get an abundant number copy of your target gene, and so in order to use the technique to detect the phytoplasma causing cassava Swiss broom diseases, the first thing we have to identify the target gene, and this has been done by colleagues. And this, you can see this the sequence of the 16S RNA of phytoplasma causing CWD, and we have to use its sequence to design a specific primer set to amplify their sequence. Designing a primer for lamb can be done either using the NCAN website from the company, and we usually use Primer Explorer 3 or 4. There's also other source of software where we can design the primer. So after we use the target gene and the software, we get this specific primer set, and then with this primer set we can use to amplify the target gene, and to visualize the positive reaction, we can use a color. We use a color indicator for a positive reaction. As you see in this slide, the H&B hydrogen atom blue is using to distinguish between negative and positive. So after a series of tests, so now we have developed an assay that you can use to test the disease sample you collect from the field. So first, after collecting the sample from the field, you need to extract the DNA using a special method. And this extraction, you have to ensure that there no contamination or no impurity is present. So after you get the DNA, the purified DNA, the next step you need to do is setting up the reaction. And because lamb is very sensitive, you need to be very careful when you are setting up a reaction. And in order for lamb to work, we need a special enzyme. We call it BAT polymerase. So after we set up the reaction, the next is we have to incubate them at a constant temperature, and we don't really need a PCR machine. We use a very simple heat block incubator. So what you need is to set up the equipment to 63 degrees Celsius and keep for 60 minutes. So there's the counting. Until it's 60 minutes, we are ready to see the result. So now you can see the result. In the top line, this we are detecting the internal control target gene from a cassava. So this one should be positive in all disease or healthy cassava. In the lower part, we are detecting the cassava switch broom disease is phytoplasma. So as you can see here, when there is this is a sign of a positive reaction. That's a blue, and then why the negative is somewhat look like a purple, and this is a negative water control, and this is help for example of an example for cassava sample, and this reaction means that the DNA quality is very good, so it supports amplification. And in the lower part now, you will see that we are detecting the target gene of phytoplasma. So this is a negative water control, it's no reaction. This is a healthy plant. This is a healthy plant. In the DNA was good, so there's amplification, but there is no phytoplasma in there. And these three samples are infected samples we collected from the field. So now you can easily distinguish between disease and healthy plant from cassava switch broom diseases. So I hope you enjoy our presentation, and we look forward to working together with you or meeting you somewhere in the future. Thank you.